Hello, everyone, and welcome back to another discussion in our flipped activities for microbiology. So, in chapter 4, what you will notice is that we talked primarily a lot actually about staining that we have done in class, or you should be doing in class fairly soon. And the staining that we do in class, in our class, and in most introductory microbiology class, is going to be gram staining. So, there's going to be quite a bit of discussion about that. So in chapter 4, entitled Microscopy, Staining and Classification, that's really a lot of what we've gone over. So in this flipped activity, that's what we're going to spend a lot of time talking about. So for my students, I don't spend a whole lot of time talking about the microscope because I feel like the book has done a really good job of explaining some of the features of the microscopes, the differences between those two classes of microscopes, between light microscopes and dark microscopes. In our class, we're not going to use anything except for a bright field microscope, but sometimes we can use some negative staining techniques. The one microscope that I want you to pay special attention to that is a light microscope that we won't use, but the fluorescent microscope because it's used for diagnostic purposes. Immunofluorescence is where we have a fluorescent dye that links to an antibody, and then the antibody um, dye combination then binds to the target, and that allows us to see it and to make it visible. So that is the one other light microscope I do want you to know what happens there. All right, so let's go ahead and move on to what you're actually coming here to see, right? So this first little blurb here just talks about what this whole staining process is, about fixing the bacteria to the slide, the wash steps, all of that good stuff. So we're going to go ahead and start on this side. And I hopefully during this time that I've been talking, or you may pause this so that you can read this. Hopefully this visual will actually allow you to see the words better if you're not one of my students and you are also watching this. So a student from a research lab is having trouble preparing a slide for examination and photographing. The bacterial slide that he has brought to you, he was prepared using commercially purchased stain. He has asked you for your help in determining what he is doing wrong so that he can change the lab protocol and continue with his project. After examining the light under oil immersion, you determine that no bacteria are present, even though the student is able to show you the culture he used to make the slide has visible growth in a liquid medium. Part A. Which of the following statements does not explain the fact that there are no bacteria present on the slide? So the first statement says insufficient heating of the slide did not drive out the thin layer of water and thus resulted in minimal bonding between the bacteria and the glass slide on there. Well, if you read that whole little blurb that I kind of skipped reading, it talks about the whole purpose of heat fixing a bacteria to a slide, how that process happens. And in none of that, nor in your textbook, does it really talk about, um, uh, well, in all of that, it actually talks about how the whole purpose of fixing those bacteria to the slide. So, insufficient heating of the slide did not drive out the thin layer of water and resulted in minimal bonding. So, the bacteria literally washed off. So, that is a possibility. Two, rinsing with alcohol during the wash step stripped the bacteria off the glass side. So, as we're reading this blurb, we understand that we use alcohol in gram staining, but in this particular simple stain, you only used one single dye. So, crystal violet, methylene blue, um, safranin, those are the only dyes that you're going to use. So there was no alcohol used. So that one there, let's put a pin in that one because I feel like that one's not really telling us the whole story there because we didn't even use any alcohol. Overheating during the fixation step boiled the water in the bacterial cells, resulted in the cells bursting. That's something that can happen if you overheat them or you char or burn your slides. And if you ever have like little brown spots on your slides, it's because you cooked it in the Bunsen burner too long, and it if its slide doesn't crack and break first, so that can happen. By not allowing the glass slide to completely dry, air dry before heat fixation, the flame will cause surrounding water to boil and damage the cell wall, and that's pretty much saying the same thing as the third bullet there. I guess I should let you all see that. So that's pretty much saying the same thing as this bullet. So the best response to this is rinsing off the alcohol. That one does not explain what happened to the student slide. So part B, You volunteer to help a student with his fixation technique and in choosing a proper stain for the project he's working on. After watching and helping the student correcting a problem with his fixation technique, you now need to determine which stains to use. The single criterion for his project is to be able to determine cell shape and size in pure broth culture after some treatment. No differentiation between 12 cell types is required, so it doesn't have to be gram positive or gram negative. So you want to convince the student that a simple stain would be his best option. The charged dyes used in simple staining will penetrate the bacterial cell wall and retained after rinsing the slide with water to remove the surplus dye. Which staining procedure would be best to use to stain the slides required for the student's project and why? So, big take homes here is that we want to stain the cells so that we can have some contrast and to be able to see the cells. We don't want to stain the capsule. We don't want to stain the background. We want the contrast to be from actually staining the cells themselves. So, in this discussion here, this is where we start talking about, and in your textbook, I'm going to see if I can find exactly where this discussion is. It starts on page 104 in the Ballman 5th edition of the textbook. And it continues on to specifically on page 106 where it talks about acidic dyes and basic dyes on there. So acidic dyes are going to be acidic as they would be found on the pH scale. So that means that they're going to have a low pH. Positively charged chromatophores are called basic dyes because they can combine with and stain acidic structures. And they can actually, the pH of them is going to be more basic. So they're going to have a higher pH to it. So keeping in mind that the membrane of a cell, and this is something you've probably learned in a introductory, an A&P course, an introductory anatomy and physiology course, or even just a cell biology class, that the resting membrane potential of any cell is typically negative. It's like negative 70 millivolts, right? So cell membranes have a tendency to be more negative. They have a more negative charge to them. So we want something that will allow us to be attracted to that negatively charged component of the cell wall. So we want something positive. So we want a positive simple dye that is going to be attracted to that negative cell membrane. So let's look at our responses. An acidic dye such as nigrosin will stain the cells a dark black coloration for easier viewing underneath the microscope. That one doesn't really work because acidic dyes actually stain alkaline structures. So that is just acidic dyes are not going to stain the cell because we have a negative and a negative charge together and they repel. Malachite green and heat will decolorize the cell for easier visualization and is a simple process. For my students, you've probably already done the endospore stain, and was that a simple process? No, there are a couple of steps in there. You had to actually put it over steam heat in order for the process to take place, and it was a thing. And then there were some students that it just didn't even work out correctly. So it's not really a simple process. There's something that's much more simpler than that. Letter C, using crystal violet followed by a rinse and grams iodine will stain the cells dark blue with large crystals. Well, crystal violet is what color? Purple. So C doesn't work for us either. But letter D, the positively charged methylene blue will be attracted to the negatively charged components of the cell wall and be retained. That is our best answer there. That is the correct answer there because it's able to be attracted to that it's attracted to that negative cell wall. So we want something that's positively charged as a dye to be attracted to the cell wall. All right, moving on to gram staining. So we talked about gram staining in chapter 3. We've performed gram staining in lab, or you will be performing gram staining in lab if you haven't already done so. Your gram staining is just something that's a really important differential stain that we use in introductory microbiology. So for the gram staining process there, what it wants us to do here is to put all these things in the correct order. And remember, for my students, you have the handout for this. For students that aren't in my class, if you're using Bauman's Microbiology with Diseases by Taxonomy by Bauman, you can find this at the Mastering Microbiology website. You can find all of these questions there. So let's go ahead and move on to this page here. So this would be the correct order for those the chart that we're looking at here. So first thing, all the cells are going to be stained purple because you put crystal violet on it. So whether it's gram-positive or gram-negative, everything on that slide is purple. The next thing that happens there is that the dye molecules are rendered insoluble because of our mordant, and our mordant was iodine. So the dye is rinsed away from most parts of the slide, but it's retained by the cell walls of those bacteria or LPS layer, the phospholipid LPS layer, outer membrane of gram negatives. Then we come through with our alcohols. So the cell walls of gram negatives are broken down, completely washed away. Gram positive cells are going to remain intact, so they're still going to be that nice purple color. But then we come back in with the saffron because our gram negatives are now clear, for if you will, or colorless, and we add that red, which stains the cells red or pink, and the color of the other cells, the gram positives, remain unchanged. So then our next question over here is concerning negative staining. Nigrosin staining, not saffron staining, of Klebsiella pneumoniae will allow for the visualization of the cell's shape and determination of the presence of a capsule. So for students that were in my class, this is the India ink we used, or computer ink, I forgot which one we're using now, that we did in our staining lab. And we remember that the background was black, that all stained black, but what we could see looked like little spaces or little rice that were embedded in this black background. And we just determined that those were the Klebsiella, and it was the capsule that we were able to actually visualize underneath the microscope. So for this question here, it is absolutely B, true. All right, so once again, we have animations, and I have not yet figured out or deciphered if the animations show up properly for us. So I want you guys to pause, and the description box will have the link to each one of these animations in order. So go ahead, pause, come on back, and then we'll go through these questions together. Welcome back. So what we're doing now that we've talked about the cell walls, the differences in the cell walls, and we've also talked about kind of how we can visualize them and how we can gram stain them. The other portion of chapter 4 is going to actually talk a little bit about using a dichotomous key, or classification of microbes. And on page 112 of your book, if you're using the 5th edition of Microbiology with Diseases by Taxonomy, Bauman's book, you're going to see just a neat little taxonomic scheme there that's referring to ticks and that kind of thing. But we're going to be more concerned with the classification of bacteria. And so since we can't really classify bacteria solely based on size and shape, they're helpful. And we can't just solely base them on being gram positive and gram negative. And once again, that's something that's helpful. Then we have to use what are called biochemical tests in order to build this dichotomous key to help us to figure out exactly what organism we are working with. So when we go here to part A, why is visualization not sufficient to properly identify bacteria? And so we have here options A, identification is only needed in clinical specimens. Well, that's not actually true. There are some things that we need to be able to identify them for other reasons, other just for clinical reasons, but to see what is normal, what is not normal microbiota. So that one doesn't work. B, not all bacteria can be seen with a light microscope. That one doesn't work either. There are some that are very small, but most bacteria can be seen with some form of light microscopy. And that's not a reason why visualization is not sufficient enough. Letter C, many unrelated bacteria share the same shape on there. There are lots of bacteria that have kind of a similar shape, but they also are have some similarities to them. So that one doesn't work. Bacteria have a limited set of shapes, and many unrelated bacteria share the same shape. So that one there is actually our best response, because we have about 3 different shapes: Bacillus, cocci and the beryllium or the spirochete. So there's really just circles, rods, and squigglies out there, and then they're not all necessarily related to one another. So we can't just look at them under a microscope and say, oh, you guys all have to be in this particular class or another. So letter D is our correct answer. Part B, what is a hallmark of a dichotomous key? And this comes straight from the animation or the video that we watch. That it consists of a series of paired statements in which only one statement of that pair applies. So it's a yes or a no question. And so for my students, we won't make our dichotomous keys based off of yes or no. We're going to use positive or negative, which means it did react, it didn't react. It is this or it's not that. But it still works the same way. They're yes or no statements. So the only one that gives us that answer, that they're yes or no statements, they're not open-ended questions, they're not only relating to biochemical process in the cells, although that's a big part of the dichotomous key structures in microbiology, but it's not the only part of it. And it doesn't only relate to the shape of the cell, and once again, that's a part of it. So letter B and part B, that is your correct response. Part C, biochemical tests are what? Remember, we're not going to look at our options there. We're going to write down what we know about biochemical tests that we learned from that video that we watched, that these are going to be tests that we use on the bacteria to help us determine what that bacteria is. And so for the responses that we have, they're the only one that looks like tests that are used to help us figure out what a bacteria is. The only one that really says that is letter A. Letter B, used to determine rate of growth. That doesn't work for that. Are visualized using microscope. Biochemical tests are biochemical tests. There's you can see the results in just a test tube or an agar plate. You don't need a microscope for them. Are the most effective way to determine bacterial shape? Well, you can't tell anything about shape or even gram staining from the results of a biochemical test. You can just really see what are the results. How do they ferment lactose? Yes or no. Are they do they produce indole? Yes or no type thing. All right, so moving on, we get some more practice with dichotomous keys, and go ahead and pause the video and watch the animation, and we'll be back. All right, so we're gonna try something here. Now that you've actually watched the video, we're gonna try to see if we can at least look at the video, even if the sound doesn't play for us properly. And
Let's practice using a key to identify.
I wanted us to look at this video simply so that we can refer back to it when we're answering our questions. So part A says, why is the unknown in this example not Pseudomonas? So I'm going to try to make this a little bit bigger here. So what we're going to do is that we're going to go to our key and look to where we have Pseudomonas. So here's that dichotomous key in those series of paired statements, those yes or no. Does it have a gram-positive cells? If yes, then we ask the next question, are the cells rod-shaped? If yes, Listeria is what we're working with. If they're not gram-stained, are they rod-shaped? No. If no is the answer, then can they tolerate oxygen? Yes. If so, then it is Listeria is what we're going with. So this whole picture here, this whole dichotomous key, this chart over here is really all of the words that are here just out in a key type form, just kind of put in like an infographic, if you will. So what we can do is we can work this key backwards. We can work this key from Neisseria. Yes, it can tolerate oxygen. No, it's not rod-shaped. No, it's not gram-positive. So we can work this key backwards as well. And we can do the same thing because remember, this side is just an example of this, the dichotomous key chart. So if we go to Pseudomonas here, we said the question is, does it ferment lactose? And then for Pseudomonas, no, it does not. So when it says, why is this unknown not Pseudomonas? Well, because Pseudomonas ferments lactose. And if we go on down through the rest of this chart here, because over here is the unknown, and then this is what the unknown is. This is what a positive result looks like. That's what a negative result looks like. Let's see if we can get to think this is the ferment. All right, so here it is, the fermenting lactose one. So here's the fermenting lactose question here. So this is what our unknown looks like. And this would be a positive result. When you get your media for any of the sugars that we use, it starts off as a red color. And then if it can ferment it or if it can break it down, then what happens is that it actually would turn the media a nice yellow color. And it can sometimes produce a gas in that little tube that's on the inside. So there's a gas that's produced there. So this would be a positive result. So this is what a positive result looks like, and this is a positive result. Since Pseudomonas does not ferment lactose, it would look like this. So if the unknown had been Pseudomonas, then it would have been this color. So letter A on part A, why is the unknown in this example not Pseudomonas? It ferments lactose. The unknown sample does. Part B, what test indicates that the unknown can utilize citrate as its sole carbon source? So over here at number 6, can it use citrate as its sole carbon source? That would be the test that we would use for that. And so for what that would look like, let's go ahead and fast forward a little bit. So a positive result would look blue, a negative result would look green, which is kind of what the media starts out as, this nice pretty green color. So when we put our unknown sample on the citrate, and since our unknown sample turned blue, and blue means yes, then that's how we know if it can use citrate as its sole carbon source. So that would bring us on this side of the key now. So for our question in B, what test result indicates that the unknown can utilize citrate as its carbon sole source? It will turn the media blue. Letter C. Part C. How would the results be different if this organism was Salmonella? So once again, we're going to work backwards on our key. So Salmonella, it produces hydrogen sulfide. So what our unknown did, so let's move it on. Move it on, move it on. So this is our unknown. So, and this is what a negative result looks like. This is what a positive result looks like. So our unknown does not produce hydrogen sulfide on here. So if the organism was Salmonella, because does it produce hydrogen sulfide? Salmonella does. So if the unknown was Salmonella, then it should have turned this color. So for our questions, it would produce hydrogen sulfide if our unknown was Salmonella. Part D, how is fermentation of lactose determined? And that was just from the video, and we also talked about this as well. And we can go backwards, back to this one here. So lactose, glucose, sucrose, there are a host of different sugars, maltose, that we're going to use in our unknown testing. And what we're going to be looking for is a color change, because this red color change is how all the test tubes show up. That's how before they're ever inoculated, you put anything in them, they're going to be this red color, reddish, pinkish sort of color. Once you inoculate them, if they can break down or they can utilize or ferment lactose, they can use that for an energy source, they're going to produce acids as a byproduct. And it's those acids that will trigger the pH indicator that's in the dye and will turn that very pretty pink or reddish color, it'll turn it a nice yellow. So the drop in the pH, because acids have a lower pH, pH is of 6 and below, the drop in the pH turns the indicator dye yellow. So letter D is your best response for that one there. So for this dichotomous key, how many questions are needed to determine that the bacteria, determine that the unknown is bacteriorides? Well, there were only 3. So there were just 3 questions that we had to get us to this point here. So the first question was, does it have gram-positive cells? No. Since it didn't have gram-positive cells, then we could go over to the other side, and then the next question is, are they rod-shaped? Yes. If they're rod-shaped, can they tolerate oxygen? Yes, they can tolerate, or if yes, then go on down to 5. Does it ferment? Actually, no, it didn't tolerate oxygen. So no, it didn't tolerate oxygen, so it was bacterioride. So there were just 1, 2, 3 questions that we had to ask. All right, so now getting back to our questions. We're going to watch another animation. So in this next animation here, and remember the link to all of these animations are going to be in the description box. We'll watch this next video. So pause me and then come back. All right, so part A, how many answers are there to a question in a dichotomous flowchart? Remember, it's either or, so there are only two, letter B. So it's either positive or it's negative. Or in the keys that we've been looking at, it's either yes or it's no. Part B, why are flowcharts useful in dichotomous keys? Um, and the correct answer for that one is that they allow researchers to visualize the relationship between bacteria. So it allows us to see how they're related to one another. So this is just information that you would have gotten directly from the video itself. And then part C, the first question the dichotomous key addresses gram stain differences. So A, we want to know, does it gram stain? So by being able to see what it gram stains, that helps us to very quickly determine and figure out which way that we're going to go down the key. Now you can build your dichotomous key any way that you would like, but typically they start with the gram staining. They start with what we call cell morphology. What does a cell wall look like? So gram staining, is it gram positive or is it gram negative? Or is it also is it cocci? What's the shape of the cells? Are they bacillus or are they rods? In pretty much every microbiology class, you're going to have a project embedded in that class called your unknown, where your professor is going to give you a test tube and you're going to tell that professor based on the gram staining process and running the results from the biochemical test that you are going to perform on your bacteria, exactly what that bacteria is. You're going to build your own you're going to build your own chart, your own dichotomous key. So for my students, we're going to actually do this as an activity in class before we have our first unknown. For other students, there are lots of different online resources and videos that will help you with how to build a dichotomous key, and I will also be talking about that later on in the semester. So next animation, go ahead and pause me and let's hear a little bit about Cindy's sore throat and her what's going on with her strep throat. So pause me and then come back. All right, so part A, treating walking pneumonia. Mycoplasma pneumonia is a bacteria that lacks a cell wall. It causes atypical pneumonia. Which antibiotic would be most effective in treating this infection and why? So, since the question tells us that this bacteria does not have a cell wall, immediately off the bat, any of these drugs or any of these responses that say that they would treat it with a drug that attacks the cell wall is going to be useless. Why? Well, this guy doesn't even have a cell wall. So we're going to look through our list and say, like, what here is saying anything about a cell wall? And if it says something about a cell wall, we have to cross it off. So A, vancomycin, because it binds and blocks the terminal 2 amino acids on the short peptide attached to NAM. When you say NAM, you're thinking N-acetylmurmuric acid. When you see N-acetylmurmuric acid, you're thinking the sugar peptide cross links, the sugar peptide combination of a cell wall. So A doesn't work. We're going to move on to B. Penicillin, because it inhibits the enzyme that cross-links peptidoglycan. No, we can't have any drug that's going to be targeting peptidoglycan because it doesn't have a peptidoglycan cell wall, so that's a waste of time and energy there. So letter B doesn't work. C, ciprofloxacin, because it inhibits an enzyme necessary for bacterial DNA replication. That one would work because it's not a drug that targets the cell wall. We know it has to have DNA because it's a bacteria, it's a living cell. It's not in its nucleus, but we do it does have DNA. But let's see what letter D says. Fluoxacillin, because it's a penicillin that resists the activity of the penicillin degrading enzymes. Well, we're not concerned with the bacteria's ability to break down the drug. We are, so it doesn't really, and if it's a form of penicillin, then it's not going to work because penicillin works by inhibiting peptide cross-links. So our best response here is letter C. Part B, many gram-negative bacteria are naturally resistant to penicillin. Which of the following would make them more susceptible to penicillin? So remember, the reason that those gram-negatives were not super susceptible to penicillin is because it had that Eller lipo outer membrane that was made of lipopolysaccharides and phospholipids. So if it has that extra membrane, it's difficult to get the drug down there to the cell wall because it's important that penicillin attaches directly to the cell wall to disrupt or inhibit those cross links between the peptidoglycan, those peptide cross bridges. So that's why the gram negatives are less susceptible to penicillin. So if we want to get penicillin to that cell wall, then what we'd probably want to do is allow those porin channels to be more permissive and allow for more drugs to get there. So A, producing a porin with a more permissive channel, that's going to be our correct response. B, we don't want to make a thick layer of peptidoglycan. C, we don't want to make penicillin degrading enzymes on there because we want the penicillin to stay intact so that it can actually attack the cell wall. And D, we don't want to produce a NAM to NAM cross-linking enzyme to which penicillin cannot bind. Well, that's doing the opposite of what we want penicillin to do, which is to bind to the cell wall and attack it. Part C, so letter A is our correct answer for that. Part C, how can a sensitive cell survive exposure to penicillin? So how could penicillin-sensitive streptococcus grow in the presence of penicillin? So what happens here is, so let's go back to what the function or the job is of the cell wall. The job of the cell wall is to prevent osmotic lysis. So if the cell wall is no longer doing its job, then that cell is going to be vulnerable to osmotic lysis. So that means that if it's in a hypertonic environment, it will have the tendency to lose water out, shrivel up, and die. If it's in a hypotonic environment, then it's going to take on too much water and it will swell up like a big balloon and then it's going to burst. In either case, hypertonic or hypotonic, the cell is going to be kaput. The cell's going to die. However, there is a happy medium. It's an isotonic environment where the solutes and the solvent concentration is roughly equal both inside and outside of the membrane. So if there is no cell wall that's there, because penicillin has degraded that cell wall for our Streptococcus species, if it's in an isotonic environment, then we're not at risk of osmotic lysis. So the cell wall doesn't have to do its job because we're in this isotonic environment. And the only one that says that is letter A, by growth in an isotonic environment. Part D, I feel like I skipped something. Oh, here we go. Part D, tetracycline is an antibiotic that inhibits protein synthesis by bacterial ribosomes. It enters the cell by diffusion. Many cells resist the action of tetracycline by making a membrane protein that pumps tetracycline out of the cell. What type of membrane transport does this resistance mechanism represent? So you remember, if you're going to be moving things from low concentrations to high concentrations, that's going to be going against its concentration gradient, and we had to use energy for that to take place. Of the two classes of transports that we've talked about, the one that's going to require energy to move things against their concentration gradient was active transport. So we want letter C, active transport. Part E. Tetracycline is, oh, did I read them? Okay, here we go. Ciprofloxacin is an antibiotic that inhibits bacterial DNA replication. It enters the cells by diffusion. Many cells resist the action of cifro by expressing a membrane protein that pumps the antibiotic out of the cell. What does the cifro-resistant protein have in common with other porins? Well, it's moving things, so it's not defensive because you're not actually eliciting an immune response. It's actually moving something. Not being structural, so porins aren't structurals. They don't help to maintain the integrity of the membrane or the structure. They don't hold things up. However, they are both moving things in one direction or another, so they are transport proteins. They're not the type of proteins, letter D, that are both enzymes. They're just moving things from one area to the next. So letter C, they are both transport proteins. So identifying microbes, letter F. Cindy was diagnosed with a rapid strep test. What kind of test is this? So because we were looking at the adherence of the antibodies to the antigens, or the antigens attaching onto various bars on the strep test, or areas on the strep test, it's what we call a serological test. So it's a serological test because we're looking at antigen antibody specifically. We're looking for those things. Okie dokie. So let's go on to our next image here. And we're classifying and sorting these cells by what structures we'd find in them. In prokaryotic cells only, they have peptidoglycan and they have that nucleoid region. Eukaryotic cells have a nucleus and mitochondria. Both prokaryotes and eukaryotes are going to have that cytoplasmic membrane. Ribosomes, although they're not the exact same size of ribosomes, they both have ribosomes and DNA. They have genetic material. All right, so that's all we have for chapter 4. The next chapter that we'll be going over will be chapter 5. And chapter 5 is going to be all about microbial metabolism. So, exactly what do bacteria or microbes, what do they eat? And how do they use that for energy? And do they use cellular respiration, fermentation only, or a combination? Is it aerobic or anaerobic? So, we'll be talking about all those things in our next chapter.